In this video, topic 1.4, we will examine the properties of biological molecules. The structure and function of polymers are derived from the way their monomers are assembled. First of all, we'll look at the nucleic acids, which our biological information is encoded in the sequence of the nucleotide monomers. Each nucleotide has structural components. These include the 5-carbon sugar, a deoxyribose or a ribose, a phosphate, and a nitrogenous base. These nitrogenous bases include adenine, thymine, guanine, cytosine, and uracil. And we'll examine later the differences between DNA and RNA in both structure and function. You can see on the right-hand side of the slide that is the structure of a nucleotide. As I had just mentioned, a nucleotide has 3 major components: the phosphate group, the nitrogenous base, and the sugar. Nucleotides, which are the monomers, are connected by a phosphodiester bond to form the nucleic acid. That is the polymer. The phosphodiester bond is a type of covalent bond. You will notice in this slide how the 2 monomers, the nucleotides, are bound together. This is a condensation reaction or a dehydration synthesis reaction. In order for these 2 monomers to come together, a water molecule is liberated between them, forming a phosphodiester linkage, or we also describe this as a covalent bond. Adding many of these nucleotides together, we end up with the structure that you're probably familiar with in DNA, the double helix. The next macromolecule are proteins. Proteins are the specific order of amino acids to form a polypeptide chain. This is the primary structure, and it determines the overall shape of the protein. Amino acids have directionality, with amino terminus, that is the NH2, and a carboxyl terminus, the C double bond OH terminus. We represent this by the amino, or the N terminal, and the carboxyl, or the C terminal of the amino acid chain. The R group of an amino acid can be categorized by the chemical properties. These properties include hydrophobicity, hydrophilic, ionic, and it is this interaction of these R groups that determine the structure and function of that region of the protein. There are actually 20 different amino acids that our body is able to utilize. Each amino acid has the same structural backbone, the amino group, the carboxyl group, but what's unique with each amino acid is that side chain. This is the way that your body takes these individual monomers and adds them together to form long peptide chains and eventually forming a protein. Similar in the nucleic acids that I just discussed, you can see that this liberates a water molecule. Therefore, this is a dehydration synthesis reaction. And in proteins, we call this a peptide bond. This is also a form of covalent. The next macromolecule are carbohydrates. Complex carbohydrates comprise of sugar monomers whose structure determines the properties and function of that molecule. Sugars be in the form of a simple sugar, such as monosaccharides. Some that you may be familiar with include glucose, fructose, and galactose. Disaccharides, di being 2, these include maltose, lactose, which is in milk, and sucrose. And they also form complex carbohydrates, and these are our polysaccharides, such as starches, fibers, and glycogen. So what is the importance of carbohydrates? Well, carbohydrates are a major fuel for our body. They also act as a building material. All carbohydrates have a very unique ratio. They always come in the ratio of 1 carbon, 2 hydrogen, 1 oxygen, or CHHO. The monomer unit is called a monosaccharide. You'll notice that the word saccharide is usually for sugar or carbohydrates, di meaning 2, and poly meaning many. Polysaccharide. Interestingly, polysaccharides act as storage in plants, such as plant starch, and in humans, we actually don't have starch, but rather we store the carbohydrates in the form of a glycogen molecule. Plants also use carbohydrates for structural support, such as cellulose, and actually in arthropods, they actually use carbohydrates as chitin, and that's actually the shell of, say, a beetle. In order to build disaccharides, we undergo a dehydration synthesis reaction. Similar to the other reactions that we just spoke about, these include the liberation of water. So once again, this is a dehydration synthesis reaction. Water is removed between the two monomers in a disaccharide molecule. In sugars, we call this a glycosidic linkage. The final macromolecule are lipids. Lipids are nonpolar macromolecules. Lipids are composed of carbon, hydrogen, and oxygen. In fact, these are long hydrocarbon chains. Within the family, there are fats, phospholipids, and steroids. Now, uniquely, lipids don't actually form polymers. However, these are big molecules made of smaller subunits. However, these subunits are not a continuous chain, such as the other three that we just discussed. So the structure of a fat molecule usually starts with this molecule called a glycerol. Glycerol is a 3-carbon alcohol, and that alcohol is joined by 3 fatty acid molecules. Once again, using a dehydration synthesis reaction to form what we call a tri being 3 glyceride. In this particular reaction, 3 water molecules are removed. In this situation, the bond that we form, once again, is a covalent bond. When we talk about fats, we call this an ester linkage.
Lipids are nonpolar macromolecules. The differences in saturation determine the structure and function of lipids. There are 2 types of lipids. The first lipid is a saturated fat. These do not contain double bond carbon-carbon. They are structurally very long, straight chains. They are mostly found in animal fats. They are solids at room temperature. So think of butter is a solid at room temperature. And the reason for this is that these molecules compact closely together. In the diagram below, you can see how a fat molecule is this box-like structure. And so when you have several of these side by side, they pack very closely together, allowing them to be a solid at room temperature. That's in sharp contrast to the other type of fat that we call an unsaturated fat. An unsaturated fat is any fat that contains at least one double bond in the carbon-carbon backbone. These unsaturated fats are commonly found in plants and fish. These oils are a liquid at room temperature. And the reason why they're a liquid at room temperature is because in the model there, you'll notice that the fat molecules do not form that nice box-like shape. Instead, what happens is that double bonded C prevents the molecules from packing closely together because one or more of the fatty acid chains have a kink in them. And that kink is caused by the double bond in the carbon backbone. Remember the differences between saturated and unsaturated fats. The other thing that's not in this slide, but that you probably know, is that saturated fats have been linked to cardiovascular diseases. Phospholipids contain polar regions that interact with other polar molecules, such as water, and with nonpolar regions that are often hydrophobic. This right here is an example of a phospholipid. You notice that a phospholipid has a hydrophilic head. Hydrophilic means attracted to water. So that end of this molecule wants to come in contact with water. The other end, the 2 tails of our phospholipid, are hydrophobic and want to hide away from water. So what that results in is the unique properties of this molecule. When you have a collection of these phospholipids, what they do is they have the propensity to form a phospholipid bilayer, and that's shown on the left-hand side of the slide there. You can see how that in your cell, the outside of the cell is water. We call that the extracellular space. and the inside of your cell is water. We call that the cytoplasmic side. And what you'll notice is that because of the unique properties of a phospholipid, what they do is they form that bilayer, such that the hydrophilic head points towards the water, whereas the phospholipid tails come in contact with other hydrophobic tails. And what they do is they push the water away. So the water is excluded. The next type of lipid are the steroids. So steroids have a common structure. These are 4 fused carbon rings. And then in addition to that, they have some other side chains. So what you'll notice is all 4 of these steroids have the same backbone structure. Different steroids are created by attaching different functional groups to the ring. The next important lipid we want to discuss is cholesterol. Cholesterol is an important cell component. It is often found in animal cell membranes, and it is a precursor of all steroids. So in other words, those steroids are essential for our function of our body are actually derived from cholesterol. So you need cholesterol in your diet, just not very much. High levels of cholesterol in the blood may contribute to cardiovascular disease.